Hello all, myself Pratima Wagh from Department of Microbiology, KTHM College, Nasik. Today I have selected Medical Microbiology MB331 for T.Y.B.Sc students. In that, our second topic is study of bacterial pathogens with respect to classification and biochemical characteristics, antigenic structure, viability characteristics, pathogenicity, as well as a pathogenesis, laboratory diagnosis, epidemiology, prophylaxis, and chemotherapy of Shigella genus. Here, we'll see in detail bacillary dysentery, that is Shigellosis, the disease which is caused by Shigella species. It is acute infectious disease of intestine caused by dysentery bacilli. Genus Shigella is gram-negative rod-shaped microorganism which is non-motile as flagellas are absent. It is facultatively anaerobic microorganism, non-spore forming, non-capsulated microorganism. They are differentiated from the closely related Escherichia coli on the basis of pathogenicity, physiology, that is, failure to ferment lactose or decarboxylate lysine, as well as they are differentiated on the basis of a serological test. In the classification, first we'll see there are various groups of this Shigella called as a group A, group B, group C, and group D. This classification is based on difference in O antigen, that is somatic antigen or antigen associated with body of that particular bacterial cell, as well as it is based on some biochemical reactions. Based on this, group A is identified as Shigella dysentery and they are labeled as 1 to 10. In group B, Shigella flexneri, that is from 1 to 6, group C, Shigella body 1 to 15, and group D, Shigella soni, that is only 1. Based on mannitol fermentation, also we can classify the Shigella species. When they ferment mannitol, then they are identified as a Shigella dysentery, while remaining 4 species of Shigella, they are unable to ferment mannitol. Shigella flexneri, Shigella boydii, and Shigella soni unable to ferment mannitol. Some general properties of Shigella, as you know, they are gram-negative cocobacillary form, and when they present in young cultures, they are facultatively anaerobic, but they can grow best aerobically also. And here on plates, you can see convex, circular, transparent colonies with intact edges, which reach diameter of about 2 millimeter in 24 hours. Shigella can ferment glucose. Exception is of a Shigella soni. They can ferment lactose on differential media, but in the later stages, that is after 48 to 72 hours, Shigella soni may show lactose positive test. Shigella form acid from carbohydrates but rarely produce gas. They may also be divided into those that ferment mannitol and those that do not. About pathogenicity, here Shigella dysentery, most severe infection causing microorganism, while Shigella flexneri, it is epidemic group and easily turned to chronic type of a disease producing organism. Shigella soni is most milder form of organism. Their virulence is because of their enterotoxin or a cytotoxin production. They are not very invasive like salmonella or like E. coli and other microorganisms, but they attach to the epithelial lining of gastrointestinal tract. They penetrate and multiply in the superficial lining of the mucosal cells, that is of a colonic epithelium cells, and there is a formation of a ulcers which is called as a inflammatory colitis. They are not highly resistant to several conditions present in gastrointestinal tract. They can survive 1 to 2 weeks in fruits, vegetable as well as in soil, and their thermal death point is 60 degrees Celsius for 30 minutes. About pathogenesis. Here, number of bacteria, that is, total number of a Shigella required to cause disease, that is, dosage, is considered important in the pathogenesis, that is, it is in the causing disease. Then, virulence of that microorganism, immunity of the person, as well as the invasiveness of microorganism. If they attach firmly to the epithelial lining, if they penetrate and multiply within the epithelial lining, then one can see symptoms of shigellosis. So, in the pathogenesis, bacteria upon entry into the gastrointestinal tract penetrate mucus and multiply in epithelial cells as well as in uh, lamina propria. There is a inflammation vessel contraction. The bacterial cell wall contain lipopolysaccharide which act as a endotoxin, and this led to endogenous pyrogenic condition, that is febrile condition. There is a formation of a superficial mucosal ulcers, and after that you can see diarrhea mixed with blood and pus starts. This leads to abdominal ache or tenesmus in the patient. Here, the normal bacterial flora which is present in the gastrointestinal tract help in the development of a certain antibodies like IgA, which prevent in attaching this Shigella bacilli in the gastrointestinal tract. So if they present abundantly and if number of microorganisms, virulent microorganisms are less, you can see there is a immediate recovery in the patient. Infection is initiated by ingestion of a Shigella, and usually the route is fecal oral route, that is contaminated food, water from that microorganism enter in the gastrointestinal tract. An early symptom is of a diarrhea. Possibly it is because of enterotoxins produced by the microorganisms may occur as the organism pass through small intestine. The hallmarks of shigellosis are a bacterial invasion of the colonic epithelium and inflammatory colitis. These are interdependent processes amplified by local release of cytokines and by the infiltration of inflammatory elements. Colitis in the rectosigmoid mucosa with concomitant malabsorption results in characteristic sign of a bacillary dysentery. There is a scanty, unformed stool which is with blood and mucus is characteristic of shigellosis. Again, I'll repeat that upon entry of microorganism with contaminated food and water, microorganism gain entry into the gastrointestinal tract. They get adhere to the epithelial lining. They enter, they penetrate in the epithelial mucosal lining. They multiply. They produces various types of cytokines, that is enterotoxin, and this leads to destruction of epithelial lining. So superficial ulcers are formed blood ooze out from that ulcers, as well as a mucus which is released from the inflamed epithelial lining, and this blood as well as mucus is now present in the stool, in the feces of a patient, which is characteristic sign of a bacillary dysentery. Here, clinical manifestation is mainly observed because of a lesion formation in sigmoid colon and in rectum. Time required for this growth of microorganism to show symptoms in the patient is 1 to 2 days, and here sometimes based on immunity of the person, load of microorganism, it may ranges from few hours to 7 days. There is acute dysentery in the patient with abdominal pain, cramps, bloating, flatulence, that is gas formation, tenesmus, and there is a continuous scanty, bloody, mucoid uh, stool in the patient, which is called as a dysentery. Other signs may include abdominal tenderness. Very rarely, fever is observed in the patient. Vomiting, dehydration, and convulsions are common in case of children. Mild cases of bacillary dysentery may last 4 to 8 days, while severe cases may last 3 to 6 weeks. And here the symptoms begin with 2 to 10 days of infection in children. The illness starts with fever, nausea, vomiting, abdominal cramps, and diarrhea. The vast majority of infections are self limited and resolve spontaneously without treatment. After pathogenesis, next and important topic is laboratory diagnosis. Here, in the laboratory diagnosis, we have to identify pathogen present in the clinical samples. So, as you know, bacillary dysentery is a disease of gastrointestinal tract. First, isolation and identification of Shigella from the feces. Here, the feces with blood and mucus is first collected uh, in freshly passed stool, possibility of presence of microorganism. So here, if that stool is not available because of a dysentery, continuous dysentery, rectal swab can be selected as a clinical sample. Here, use of a buffered glycerol saline holding solution is also used for rectal swabs. After this, inoculation of stool sample on various media, that is isolation of a Shigella. Commonly used primary isolation media include McConkey's agar, Hectoin enteric agar, and SS agar, that is Salmonella Shigella agar. Here, aerobic incubation is widely used to inhibit growth of a anaerobic normal flora. These media, McConkey's media as well as Hectoin enteric agar, contain bile salts, which inhibit growth of other gram-negative bacteria, and also pH indicators to differentiate lactose fermenters, that is coliforms, from non-lactose fermenters. This organism is lactose non-fermenting microorganism. That is why on McConkey's agar, colorless colonies appear. Various biochemical tests are used for identification of a Shigella. The important one is catalase test, which is positive, and oxidase test, which is negative. Second to this is IMVIC test. This organism is indole positive, so indole production test is positive, while methyl red test also it shows positive, and Voges-Proskus test negative. So first two tests they are positive from IMVIC, while remaining two tests they are negative. that is citrate utilization test. So by using Simon's citrate, one can perform this test. Then urea hydrolysis test is used for production of a urease enzyme, which is negative for Shigella. Various sugar fermentation tests are also carried out. As you know, this organism is lactose, shows lactose fermentation test negative, except Shigella soni, which is late lactose fermenter. Remaining three species of Shigella, they are negative to that within 48 hours. Dextrose fermentation test, it is positive, that is they can utilize dextrose and produce acid as well as a gas. Maltose fermentation test, again acid and gas is produced by microorganism. Mannitol is fermented by three species of Shigella. And lastly, sorbitol fermentation test, which is again positive. A liquid enrichment media is also used for laboratory diagnosis. Hajna gram negative broth, which is liquid enrichment media. It is inoculated with stool specimen and then subcultured on various selective media like Salmonella, Shigella, agar. Salmonella is motile, Shigella is non-motile microorganism. After incubation in liquid media, on primary isolation media, when you inoculate it and incubate it at 37 degrees Celsius, colorless non-lactose fermenting colonies appear. And after that, these colonies can be stabbed into tube slants or a Klingler's iron agar or a triple sugar iron agar. Here, Shigella species produce an alkaline slant and an acidic but with no bubbles of a gas in the agar. This reaction gives a presumptive identification. So after this, requirement is of a slide agglutination test with antisera for serogroup and serotype for confirmation of the identification. After this, there is a next important part in study of dysentery is its epidemiology. As you know, source of infection is patient itself as well as the carriers. During recovery, patients also shed microorganism in their stool. They are called as a convalescent carriers. Some are chronic carriers. They also shed microorganism from 6 months to 1 year in their stool sample. Route of transmission is fecal oral route, and here the susceptible population, that is with very less immunity, they get infected upon entry of few bacterial cells with contaminated food or water. Here we have to study some epidemic features of this disease. Mainly in summer and fall, you can see outbreak of the disease. And Shigella flexneri, soni, and dysentery, they are most common cause of shigellosis. The group of uh, people which is involved in this is younger children. Shigellosis is endemic in developing countries where sanitation is poor. That is, few cases are observed uh, in the population again and again. Typically, 10 to 20% of enteric disease and 50% of bloody diarrhea or a dysentery of a young children can be characterized as a shigellosis. And prevalence of this infection decreases significantly after 5 years of life. In developed countries, single source food or waterborne outbreaks occur sporadically, that is, one or two cases in the population after long period. Pockets of endemic shigellosis can be found in institutions, in mental asylums, in nurseries, and in remote areas with substandard sanitary facilities. So, in jails, as well as in military barracks, you can see such a type of outbreaks, mainly during wartime. Humans are the primary reservoir of Shigella species, with captive primates as an accidental host. Means, in monkeys, upon injection of this Shigella, one can see pathogenicity. In developing countries with prevailing conditions of inadequate sanitation and overcrowded housing, the infection is transmitted most often by the excreta of infected individuals via direct fecal oral contamination. Flies may contribute to spread from feces to food. The most common species, Shigella dysentery and Shigella flexneri, are most virulent. In developed countries, sporadic common source outbreaks, mainly involving Shigella soni, are transmitted by uncooked food or contaminated water. Direct fecal oral spread can also occur in institutional environment such as child daycare centers, mental hospitals, and nursing homes. The incidence rate of bacillary dysentery varied greatly according to age groups, with the highest rate among children. Despite an overall decrease in incidence rate over the time period of study, number of countries in that northwest China, that is Tibet, Gansu region is included, Beijing and Tianjin consistently had the highest incidence rate of a bacillary dysentery. In the laboratory diagnosis, as you know, here blood picture is observed to find out WBC count, as there is an increase in the level of a WBCs. But very rarely involvement of a blood for presence of microorganisms itself. There is increasing neutrophils, but at the same time, here primarily stool examination plays important role in the laboratory diagnosis. In the direct microscopic exam, presence of a gram-negative rod-shaped microorganism, but as it is confusing, as normally also number of a gram-negative bacteria present in the stool which are rod-shaped, a test is for presence of microorganisms in the whatever mucus present with blood cells and bacterial culture using this specimen. Here bacterial culture by using McConkey's media or Salmonella Shigella agar to find out etiological agent from stools. After this, biochemical test to find out whether the organism causative agent is Shigella or not, they are carried out. Sigmoidoscopy is also used to find out shallow ulcers present in the sigmoidal colon, including scar formation in the same area. Treatment is initially starts with oral rehydration as there is a dehydration because of a continuous diarrhea or a dysentery. This one has proven effective and safe in the treatment of acute diarrhea. Antibiotics are not widely used, but as it reduces the average duration of illness from approximately 5 to 7 days to 3 days, and also it reduces the period of a Shigella excretion after symptoms subside, it is used in the treatment. So ciprofloxacin, ofloxacin, levofloxacin, azithromycin are used in the treatment of a shigellosis. Severe dysentery is treated with ampicillin, trimethoprim, and sulfamethoxazole. In patients over 17 years old, 4 fluoroquinolone such as ciprofloxacin is widely used. This is disease where recovery rate is very high without treatment as body develop immunity against this microorganism very quickly. In the prophylaxis, provision of safe and abundant water and effective feces disposal is included. These public health measures are at best long range strategies for control of shigellosis in developing countries. Important one here is prevention of fecal oral transmission. So wash hands properly with soap and water before eating or handling food and after using the toilet. Cook food through thoroughly before consumption as high temperature kills this microorganism. Poor hygiene and sanitation increases the risk, so maintain the hygiene. Do not eat raw vegetables. Use boiled water. Do not use contaminated ice, that is, unless it has been made from purified water. Vaccines are not currently available. So, here, prevention of fecal oral transmission is one main preventive measure in the prophylaxis part of shigellosis. Education of all residents to actively avoid fecal contamination of food and water and to encourage hand washing after defecation. Encourage mothers to breastfeed infants. Promote use of oral rehydration therapy to offset the effects of acute diarrhea. Encourage mothers to provide convalescent nutritional care in the form of extra food for children recovering from diarrhea or dysentery. Convalescent means during recovery, treatment is of a nutrition to the children. So, this is all about shigellosis caused by various species of Shigella. Thank you very much.